Yeah, so I'm not going to really answer the question that the title suggests, but we'll talk about reasons why we might think about genetics and with BPD. This is a question I get asked by our clinical teams quite often about, should we be doing some genetic testing, and so we'll kind of walk through some of that. So I have no disclosures to declare, and I'll not be presenting any off-label use of drugs. So this is just a quick overview. We'll just provide some background and some animal models as well as human studies that suggest there may be some genetic pathways involved, but then really talk about some of the limitations of the studies. I'll talk very briefly about a project that we're just getting going here to try to answer some of these questions. And then at the end, we'll just talk about some of a framework in which to try to determine whether to test a baby. So I think the first and biggest question is, what is BPD? We talk about it like we really know what it is, and yes, it's need for respiratory support at some defined age, but when it comes right down to it, we really know that BPD is just a descriptive term and not an actual diagnosis, and does not imply any particular mechanism of disease. When we think about all the different mechanisms that could result in the final common pathway of a descriptor of BPD, multiple influences are involved. And, first and foremost is development. And so, almost all babies who develop some form of chronic lung disease have been born prematurely, and so that's certainly a significant contributor. Obviously, environment is a very important contributor as well, not only the prenatal environment, but also the postnatal environment. They can be modified by antenatal steroids, infections, prenatally, and then postnatally, and then the type of respiratory support or duration of respiratory support, all of which may provide some environmental influences on the development of BPD. I think this is an overlooked aspect, systemic disease. And so systemic disease that results in a predominantly lung disease phenotype that may have multiple different causes. And, I typically like to think of prematurity as a systemic disease, since we know that in general, multiple organ systems are involved. And so really just not overlooking the possibility that there are non-pulmonary mechanisms for disease that result in a predominantly pulmonary phenotype. And then there's the question of genetics, and we know from a lot of the work that we've done and others subsequently how the surfactant associated genes result in neonatal lung disease, interstitial lung diseases in childhood, in some cases idiopathic pulmonary fibrosis in adults. But those are the most clearly defined genes that lead to a predominantly respiratory phenotype. What also complicates the genetics of BPD is that the genes during the course of development have gestational age dependent expression. And so a gene that is active at, say, 24 weeks of gestation and may be mutated if the baby is born at term may never have any type of phenotypic expression. Whereas if a baby happens to be born at the specific time that that particular gene is most active or exerting its influences, then there may be a phenotype that results. And so that complicates the whole field of trying to identify genetic specific genes that may contribute to the phenotype of BPD. So, and also, as I always like to say, is that, you know, when somebody puts up a slide that there's just so many different pathways, and the more complex the slide is, the less we know about the particular disease or phenotype that we're trying to describe. So BPD is certainly one of those disorders. So let's look at some evidence or some data that have suggested but not confirmed that BPD might be a genetic disorder. And these go back to several twin studies that have been done, looking at concordance or discordance of the phenotype in twins. That suggests that there may be some heritable component to BPD, not necessarily a genetic component, but at least a component that's inherited. And so 3 of these studies, the Parker, Bandari, and Lavoie studies, suggested there might be a heritable component to BPD. These all looked at populations that were of different birth weight or gestational age, different definitions of what BPD might be, but they found that these particular cases might have a genetic contribution of anywhere from 50 to 70%. So these were so these studies suggested, but by no means confirmed. They also did not directly measure zygosity, so used either placentation as the definition. So we don't have specific evidence that these were monozygotic twins necessarily. More recently, though, Rich Parad in Boston had a cohort that was really the most premature of all of these studies, using this particular inclusion criteria and this the 36-week definition for BPD, and did not find any evidence of heritability. So I think the jury is still out as to whether there is a heritable component to BPD. So as I said, lots and lots of circumstantial evidence as to whether there might be some genetic component. And you know, we're all familiar with identifying or taking care of patients who look identical, not necessarily related, but they look identical from a gestational age standpoint. A reason for the reasons for delivery, birth weight, et cetera, multiple factors phenotypically, but they have completely disparate outcomes. And so, you know, we know the data that about 40% of extremely low birth weight babies develop what we call typical BPD, or what I'll call typical BPD by whatever definition you want to define it. But on the other hand, 60% do not develop, quotes, BPD. We also know that assigning a description of BPD doesn't necessarily predict the longer term outcome. And so data that we were involved with the Prematurity and Respiratory Outcomes Program, where we enrolled babies at 28 weeks and less of gestation. It was an observational study, followed them out to a year. Of the babies with moderate to severe BPD at 36 weeks by typical definitions, 25% of those babies did not have any evidence of persistent pulmonary morbidity a year out. But conversely, 40% of the babies without the definition of BPD did have some persistent pulmonary morbidity, and this was defined by chronic medication use, recurrent wheezing, hospitalizations for respiratory disease, and so on. Mike O'Shea and his group in North Carolina with the LGAN cohort. These were babies who were born in the early 2000s, a 10-year follow-up. 40% of those who were diagnosed with BPD had asthma, but then on the other hand, 36% of those without BPD had asthma. So just applying the definition of BPD does not necessarily predict the long-term outcomes. And again, is this evidence that there may be some genetic influences? Very, very circumstantial and by no means conclusive. A lot of circumstantial data comes from animal models, and I'm not going to go into the multiple publications and studies that have been done looking at various animal models of PPD. We do know that exposing mice, rats, sheep, other animals to hyperoxia in the immediate neonatal period leads to a histopathology that resembles what we call BPD in terms of alveolar simplification, decreased vascular surface area, plus minus inflammation, and so on. Multiple exposure protocols with or without intrauterine inflammation lead to this particular phenotype. And there have been then multiple examinations of various genetic genes, genetic pathways, including inflammatory mediators, TGF-beta, matrix metalloproteinases, growth factors like epidermal growth factor, FGF, insulin-like growth factor, various antioxidant molecules, and then vascular signaling molecules like VEGF or angiopoietin. So, disrupting a single gene or a couple of genes in a pathway through genetic engineering technology has suggested there may be some influence of some of these genes. Also, conversely, studies that have tried to replace or overexpress some of these molecules have led to a mitigation of the phenotype in these animal models. Again, suggesting there are potentially genetic influences that may be playing a role. There have been multiple studies in humans looking at candidate genes or gene panels, single genes with either next generation sequencing, exome sequencing, genome sequencing, or various array panels, GWAS panels, looking at 1.5 million genetic markers across the genome. Many studies have looked at common variants, some have looked at rare variants, some have looked at the combination of both. And there are some genes and gene pathways that have potentially come out. People have looked at single genes. The SPOT2 was a gene of interest about 10 years ago. The surfactant genes are certainly of interest, and here's some data that we generated. This was again from the prematurity and respiratory outcome program cohort. We were able to do exome sequencing on about 400 of these babies, and so we looked at pathogenic variants, potentially pathogenic variants in the ABCA3 gene, surfactant protein B and C, and the thyroid transcription factor gene. And we had, again, these were just all comers. We defined affected as requiring significant respiratory support of greater than 2 liters per minute at 36 weeks, and unaffected were those who did not require that degree of respiratory support. And we did find potentially pathogenic variants in some of these genes. Now, the ABCA3 gene has gotten a lot of interest. The ATP binding cassette member A3, this transport is expressed on the surface of the lamellar body, transport phospholipid from the cytosol into the lamellar body for the final assembly of the surfactant complex. And we found that in that cohort, about 5% of the babies who were, quotes, affected had a pathogenic variant in ABCA3, but then about 3% of the babies in the unaffected group had a pathogenic variant. And I must say that this is consistent with some of the population-based studies that we did about 15 years ago, looking at the frequency of pathogenic variants in the general population. And we found in those studies that about 3 to 5% of the general population carries a pathogenic variant in ABCA3. Remember, ABCA3, we think, is primarily expressed as a recessive disorder, so you need 2 copies. All of the individuals we identified here were monoallelic for pathogenic variant in ABCA3. There's some data we generated quite some time ago, as well as some emerging data that suggests there may be some babies who seem more susceptible to respiratory disease with a single ABCA3 variant. But these were all babies who were born at 28 weeks or less, and like I said, either had a BPD-like diagnosis or not. What was a little bit surprising, and we haven't had a chance to really pursue this observation yet, is that we found pathogenic variants in the SPB gene in 4 individuals. This is an extreme, these pathogenic variants are extremely rare in the general population, as noted in the unaffected group here. So I'm not quite sure if this is just a statistical aberration or if there's really something to this. Again, this is typically expressed as a recessive disorder, meaning that you need biallelic variants. We did find 1 pathogenic variant in surfactant protein C in 1 individual, and then we didn't find any in the thyroid transcription factor gene. So the bottom line is there are pathogenic variants in the genetic background of premature babies who may or may not have BPD. But when we looked at a little bit closer at their clinical presentation, just from the clinical data that we had, we really did not identify any difference in what could be classified as disease severity by most duration of ventilatory support, type of ventilatory support, etc., or in the 1-year respiratory outcomes in the babies who did or did not have pathogenic variants. So these babies would not have come to clinical attention, at least from what we can glean from the clinical data that we collected through the prop. So I think the jury is still out. I would say that right now I'm not overly enthusiastic that about surfactant genes contributing in any significant way to a phenotype of what we call BPD. Like I said, there have been many other human studies that have been performed using a variety of different platforms and so on. And many of them point to similar types of pathways. So again, like the animals, inflammatory pathways, growth factors, structural genes, antioxidants, and vascular signaling. But as many studies have been done and as many patients have been evaluated, none of the findings have been replicated in any subsequent study, either at the variant level or the gene level. And so, I think that, again, we have some circumstantial evidence that there may be some genetic contribution, but again, these haven't been replicated. Now, there are a lot of reasons for that, and I think all of the human studies to date have really had significant limitations. First of all, populations have all been different. So in general, the studies have not taken into account ancestry. So there may be varying population genetic admixture in the various studies, and that may be one reason that there hasn't been replication. European studies may have a different ancestry than studies done in the US, etc. Sample size is always a limitation, especially with the genome-wide evaluation. And so many of these studies just didn't have sufficient sample size to reach any kind of statistical significance. All of the studies have used a different type of platform, like I mentioned before, SNP arrays versus exome versus genome. There have been some studies that have done use multi-omic approaches, so genomics and mixed with transcriptomics or genomics with proteomics. Again, none of these studies have replicated again on the at the individual gene or the individual variant. But like I said, some general pathways are suggested. Now, another major reason that these studies have not been replicated is that BPD may not be a genetic disorder at all. And I think we have to keep that in the back of our mind as we do any of these studies. Despite all of those limitations, the biggest limitation, and one we have not been able to overcome as yet, is the definition of the phenotype. And again, like I said, we're describing BPD based on the treatment that's being provided at 36 weeks of age or whatever gestational or chronologic age you want to do. So it's a very imprecise definition. It is not a phenotype. And so I think that because it's so imprecise, we really don't have a pure phenotype by which to do gene by phenotype association studies. And I think that's the major limitation. So how can we really think about digging in a little bit deeper? And so there's been some interesting work over the last several years to really try to define subphenotypes or endotypes. And most people use those terms interchangeably. It's endotype implies a mechanism. Subphenotype is still a descriptive term. But if we think about subphenotypes, does that allow us to get a little bit closer to defining mechanism? And Ed Shepard and the group at Nationwide a number of years ago studied infants with severe BPD and did infant pulmonary function tests and described three subphenotypes. So an obstructive subphenotype, a mixed, a restrictive phenotype, and a mixed obstructive restrictive subphenotype. That suggests you might be able to start parsing out subpopulations of individuals by which to more precisely stratify the phenotype and do some type of association study. Wu et al. In Philadelphia a couple of years ago did a retrospective analysis of babies with severe BPD who had CT scans, CT angiograms, echocardiograms, and a subset of whom had bronchoscopy, and tried to again further refine the subphenotypes. And they described subphenotypes of parenchymal disease, pulmonary hypertension, and large airway disease. And so this was the first study that really started to kind of acknowledge at least a vascular, potentially vascular component to this. And then Piero et al. in Spain had a review article a couple of years ago where they really laid out kind of nicely, I think, a nice theoretical framework for looking at these BPD subphenotypes. So a congestive phenotype with pulmonary edema, peripheral airways phenotype, central airways, vascular, interstitial, and parenchymal. And these are all really nice kind of buckets to think about. But I also think that the reality is that most of the patients that we see don't really fit nicely into one of these categories, and there are a lot of mixed phenotypes. But I do think that this is a nice way of thinking about the type of, quotes, BPD, the types of respiratory insufficiency a baby might have at a particular time point that might then allow us to think more mechanistically about their type of chronic lung disease. There's been a lot more attention to the vascular phenotype, and Steve Abman and Pete Moraney have done a lot of work both at the animal model level as well as human level to try to describe some of these vascular subphenotypes. These are some data that we generated again through the prop. Phil Levy was a fellow with us at the time, and at the time looked at what were at that time some novel echocardiographic measurements such as pulmonary acceleration time, right ventricular strain, tricuspid annular excursion, etc. And what we found at that was these are all, again, babies who were born at 28 weeks of gestation or less. We did serial echoes on them at birth or shortly after birth, 32 weeks, 36 weeks, and then again at a year of age. And what we found was that, first of all, looking at babies who were born at term and healthy, their measurements were distinctly different from the subsets of babies who were born preterm. The preterm babies with chronic lung disease had various echocardiographic measures that were clearly different from the term babies. But then there was this group of preterm babies who did not have any chronic lung diseases by the various definitions, who kind of fell in the middle, who demonstrate that there may be some suggestion of a vascular component. And then Kara Goss and her group have studied adolescents and young adults with echocardiography and stress tests, etc., and have also found persistence of some of these findings, even in individuals who did not have BPD or in the neonatal period. And so, it does again suggest, there are some of these differences in cardiac expression of prematurity that might suggest some type of potential genetic contribution. Also from the prop where we did the exome sequencing on the babies, we did this was 150 of the babies with the most extreme respiratory phenotypes at 36 weeks. So babies who required no respiratory support at all and had not required any respiratory support for several weeks before 36 weeks. And then a subset who required the most significant respiratory support at 36 weeks. We did many different statistical approaches to this to try to understand, could we see replication among the various statistical approaches? And then we also cross-referenced what we were finding at the genetic level with what was found at the transcriptomic level. From a series of babies from Tom Mariani and Gloria Pryhuber's group, where they did transcriptomics of babies who died with severe BPD. And we cross-referenced the upregulation or downregulation of genes and pathways with what we were finding at the genetic level. And a couple of interesting things came out, none that, again, reached the genome-wide statistical significance, but they're intriguing enough because I think that they might shed some light on some of these subphenotypes. And so, and they seem to also align with some of the other studies that had been done previously. So in blue, we have epidermal growth factor signaling, ERBB, which is another member of the epidermal growth factor group, and neuregulin. So these are all members of the EGF family of genes that lead to structural development. There were animal models knocking out ERBB4 that resulted in a BPD-like phenotype in mice. So that's tantalizing evidence. Certainly no clear confirmation, but again, consistent with some studies that gone along, and also consistent with something that we might see phenotypically with the structural development of lung. I mentioned the cardiac findings, and so two cardiac pathways came up and were actually unique to the babies with BPD, and that's cardiac hypertrophy signaling and factors promoting cardiogenesis in vertebrates. Again, these pathways are very, I don't want to say generic, because there are a lot of type of housekeeping genes that overlap and are expressed in multiple different pathways. But again, given this vascular phenotype, I think it's interesting that these were a couple of pathways that came up. And then most intriguing to me, at least, is a couple of the endocrine pathways. So the gonadotropin releasing hormone signaling pathway. We know that there's this male-female difference in the epidemiology of what we call typical BPD, and so could that be a signal? And then corticotropin releasing hormone signaling, and I think this is, steroids are a never-ending and very hot topic in children with BPD. Tamara Lewis a number of years ago, using the TOLSERV cohort, identified individuals with variants in the corticotropin releasing hormone gene were less responsive to steroids. So I think that's another potential pathway for further investigation.
I'm sorry to interrupt you. Are you trying to share your slides? For some reason, I'm not seeing the slides being shared anymore. I don't know if I did something. I apologize. You have to reshare.
As I'm screen sharing.
Okay, it might just be me.
I can see the slides.
Okay, so then it's just me. Okay, sorry.
Okay. Can you see my pointer when I move it around? Apparently not.
Yes.
Yes.
Oh, okay. All right. I think, these are all interesting, circumstantial, but I think it also suggests that we could potentially use genetics as a tool to refine endotypes. And I'll just describe very briefly a study that we're just getting underway. We just got the NIH funding, and we're just putting all the pieces together. And called long-term endotypes of prematurity associated respiratory disease. And so since every big study needs to have an acronym, we use the acronym LEOPARD. And so, so hopefully you'll be hearing a lot more about the LEOPARD study going forward. But our overall hypothesis is that the BPD endotypes can be defined by mechanistically linked gene pathways. And I'm not going to go into a lot of detail, but we're going to use a lot of data that are existing. We're enrolling babies who are enrolled in a clinical study in the neonatal ICU in the neonatal period. And then these who have existing clinical and genetic information from the neonatal period. And then they're still being followed through some mechanism at school age and above. And so we'll be bringing a subset of them back. These will be babies who are, well, now children who are between 5 and 15 years of age, obtaining some new clinical data, but also doing spirometry, 6-minute walk tests, and echocardiography. And then linking all of these clinical data with the genetic data. And we'll do a typical genetic association study with various predetermined phenotypes, a vascular phenotype, an asthma phenotype, etc. But then we'll also use machine learning techniques to do unbiased or more agnostic phenotype definition, and then do the genetic association study using these unsupervised endotypes. But then the interesting thing that we'll be doing is doing what is loosely called reverse genetics. And so we'll identify gene pathways that are expressed throughout this group, and then use the gene pathways to define the clinical phenotype at school age. And by doing this, see if we can start to understand, again, more mechanistically defined endotypes in this case now, using genetics and getting to mechanism by which then we can start to, in the long run, go back to the neonatal period and maybe develop gene panels to look at the types of the genetic pathways that might be operative in these patients. We're also going to, there are lots of publicly available genomic databases for asthma and COPD, and we'll take the data that we have and see if there's any shared genetic architecture with those asthma cohorts or with the COPD cohorts, to see, number one, are there commonalities? And an emerging concern of babies who are born extremely prematurely is, are they going to develop COPD when they're 30, 40, 50 years old? And so this could lend some credence to that particular argument. So I won't go into any more detail about that. Like I said, we're just assembling the infrastructure at this point. And so, but it's an exciting new way of thinking, and I'm hoping that by doing so, we're ultimately able to eliminate the term BPD and really talk more about specific mechanistic pathways that lead to specific phenotypes. So to get to the ultimate question is, should we be doing clinical genetic testing for BPD? And I think there are quite a few questions that we need to ask ourselves, first of all. And the main question is, how are we going to use those results if we get them? And so will they be actionable clinically? How will a positive result be useful? And vice versa, will a negative result be useful? And really, truly, what does a negative result mean? And I'll get into that in a little bit. Anytime, I'm sure everybody online here has done some type of genetic testing, and you get back a variant of uncertain significance. you are going to get variants of uncertain significance as a return of results. So how will that be interpreted? How does that help you? How does it not help you? Does it confuse the family more, and so on? So I think, before even thinking about doing the genetic test, I think these are questions that need to be asked. And not to be dismissed at all are the potential for secondary findings. So these are the 80 some genes that the American College of Medical Genetics has identified as being potentially actionable, the BRCA genes, Huntington's genes, etc. And so what are you going to do with those? And so the genetic counseling before doing the testing needs to really address these. Families can opt out of getting that information back, or they can opt in. If they opt in and you find something, what are you going to do about it? So there's a lot of thought that has to go into determining whether you're going to even do a test to begin with. They will, okay, let's go ahead and do the test. So what tests will be done? And so there are a variety of tests. Will you do a microarray? What information will a microarray tell you? Well, it'll tell you whether there are copy number variants across the genome that may or may not be associated with the phenotype. Will you do a specific gene panel? Many companies have respiratory disease gene panels that may have anywhere from 50 to 100 genes on them or so. So I think determining what test will be done, and then identifying, every lab has different reporting criteria. So some will report out variants of uncertain significance, some won't. Depends on what gene it's in, whatever, and so on. So really understanding what will and will not be identified, or what will or will not be reported. I think it's important to know that when we're doing clinical or medical exome or genome testing, we're not testing the entire exome. We're not testing the entire genome. Yes, the data are being run on a whole exome platform, or being run on a whole genome panel. But typically the labs will report out known or novel variants in genes that have been previously associated with either a general phenotype, or if you're doing a gene panel, that have been limited to a specific phenotype. And so, for example, developmental delay, what they'll do is run the sequencing. They'll look at the genes that have been associated with developmental delay. Are there any variants in your patient sample that have been reported previously, either specific variants or specific variants in specific genes, and so on. And so you're only getting the data that for which there's pre-existing knowledge, i.e. a gene that has been clearly linked with a disease type of phenotype. Then there's the extended exome or genome, where they'll report variants in genes that have been associated with disease, but not necessarily the specific phenotype. And that ends up being about 4,000 genes. These are some data from the online Mendelian Inheritance in Man, OMIM, which is one of my favorite websites. And this just shows that over time, the number of phenotypes with a known molecular basis, so that's the dark circle, and that's about 7,500 phenotypes have been defined that have a known genetic association or potential genetic mechanism. In contrast, there are about 5,000 genes that have been identified that cause a specific phenotype. And so this is what I mean by when you're doing a genetic study, an exome or a genome for clinical purposes, it's really being classified and reported against this existing database of literature. And so novel phenotypes, novel genes that have not ever been associated with a specific phenotype are not going to be reported. And so you're really looking at, there are about 20,000 genes in the genome. You're looking at about 15,000 genes that are not being interrogated at all. And so the clinical exome, or the testing that you send off, is only useful for what's already known in the literature. It's not for discovery. And so that's the point of this slide, is it does not provide for gene discovery. And so it's important to know that, and it's important to know that if you get an inconclusive or uninformative result from the genetic testing, it doesn't mean that there's not a genetic contribution. It just means that the genetic description or genetic mechanism has not yet been identified. So knowing that, then I think the next question is, if you're going to go ahead and do the genetic testing, if it's negative, could the results be informative from a research standpoint? And so again, depending on the lab, depending on the informed consent, etc. Since all of these tests, the exomes and the genomes, are run on a whole exome or whole genome platform, the primary data are available. The question is whether the lab will release it for research purposes. And for that, then you need to get specific consent from the family. And if you have that, then a lot of times they'll release the primary data for you to do that, do a deeper investigation. And then you need to have the resources for doing the further investigation. And so, I tell our fellows this all the time, that, every patient for which you have sent off a genetic test that either has a variant of unknown significance or the testing is uninformative, that has the potential to be a career-making patient, because if you have the resources, you have the mentorship, etc., to really do that deeper dive, to interrogate the rest of the genome, then do the hard work to not only identify variants, but then start to do some validation, either in cell culture and animal models, etc. It's a very, very labor and resource intensive investigation. But I think if you think that there's something sufficient and substantial for research, and you have the resources available, then I think it's certainly worth considering going through getting the genetic testing and then going through the process of getting consent from the families. So when might we consider genetic testing? Certainly if there are other affected family members, I think that goes without saying. I think that can potentially be extremely informative, and especially if, for your own purposes, if there aren't any genes that have been identified previously with this phenotype, this offers you the opportunity to build your career. Extreme phenotypes, I kind of put a question mark around that. I get asked all the time, well, this baby was born at 32 weeks and had the worst respiratory disease I've ever seen in my life. Yeah, I think that's worth more of a consideration. Again, I think you have to go back and ask yourself what you're gonna do with the data, and especially what you're gonna do with it if it's inconclusive. And then certainly if there are unusual manifestations, so, you know, do you have a systemic disease with a primary pulmonary phenotype that could potentially be informative? So I haven't answered the question, but I hope that at least this provides a framework for thinking about it when to go through with genetic testing. So in conclusion, the genetics of BPD have been elusive. And I think the main reasons are it's an imprecise phenotype, and it may be that it's not a genetic disease per se, but there may be some genetic susceptibility to developing that phenotype. I think identifying or unmasking subphenotypes or endotypes will certainly be more precise. How can we do that? And I think really looking beyond the immediate newborn period and into that school age or even beyond, to really see if there might be more specific mechanisms or more specific phenotypes that become manifest. And then finally, I think that there may be certain circumstances for clinical genetic testing for quotes BPD, but I think the state of the art is not quite there as yet. And I say that as somebody who would love to do genetic testing on every baby who comes through the NICU. But I think we'd have to be careful about that. Lots of collaborators, the top ones are all those who are going to be involved in the Leopard project. My collaborators in the prop grant, and happy to answer any questions.
Great, thank you so much. This was great. Does anybody have any questions? Oh, I do have one, sorry. Has epigenetic analysis revealed any influence in addition to genetic predisposition to BPD?
So far, no. There has been some work with epigenetics, and I think that's a very important approach to try. There are a lot of challenges associated with that. Again, the biggest challenge is the phenotype description. That's going to underlie every type of approach that we try to take to this. But I certainly think that there's a role for epigenetics. The challenge with epigenetics is, what tissue. Epigenetics is very tissue specific, and so what tissue are you going to be using to do the investigation? At what time point are you going to be using, obtaining the samples, et cetera? So there are a lot of caveats to that, but it's certainly going to be an important piece of our armamentarium.
Any other questions? That was the only one that was chatted in. Anyone could feel free to come off of mute and ask a question directly. Dr. Hamvas, you mentioned machine learning and other things that might be helpful with looking at genetic markers of disease. What experience do you have with that, and what recommendations do you have for those of us who know very little about it, and how to approach it with caution and judicious mindset about, the ethical implications for using this?
Yeah, well, I fall into the same category as you. I'm one of these people who say the words but don't know exactly what I'm talking about. So that's why I have some great collaborators who know a lot more about that. Yeah, I think it remains to be seen how that's going to help inform us in the long run.
I think if it helps with big data sets and being able to look at data, large amounts of data, I could see the utility, but also I'm just as wary about it as well.
Yeah.
Well, this was great. Thank you so much. And it really kicks off our fall season looking at ventilatory strategies for babies who are really high risk for BPD. So it's great to have the framework to approach this as we think about ways to continually improve the care we deliver. So thank you so much for joining.
My pleasure. Feel free to send any questions my way.